Hello, this is the first part of Lab 6, Passive Diffusion, the movement of solutes across a selectively permeable membrane, by Victoria and I. Okay, for a brief introduction, there's the plasma membrane, which is made up mainly of phospholipids. It contains two layers, or leaflets, hence the name phospholipid bilayer. It also contains cholesterol, proteins, and carbohydrates. Each phospholipid contains a polar head and a nonpolar tail. Phospholipids are amphipathic, which means that they have both hydrophobic and hydrophilic properties. The hydrophobic part are the tails, and the hydrophilic parts are the heads. It separates the intracellular environment from the extracellular environment in a cell, in respect to a cell. And it's selectively permeable, which means it only allows certain molecules to pass through. This is a picture of the plasma membrane. Here we have, these are proteins. This is a glycoprotein because it has a carbohydrate attached. We'll get to that later. These are cholesterols. This is one leaflet, and there's two, so that's the phospholipid bilayer. And this is a glycolipid because it has a lipid attached to it. I'm sorry. It's a glycolipid because it's a lipid with a protein attached to it. A carbohydrate attached to it. I know this, trust me. And this is the chemical makeup of the phospholipids. As you can see, the tails are nonpolar and the heads are polar. So first we'll talk about cholesterol. The main function of cholesterol in the phospholipid bilayer is fluidity. The more cholesterol, the more fluid the lipid bilayer is due to more double bonds. This makes the bilayer unsaturated, which is liquid at room temperature. And one example of this mechanism being used would be if an organism lived in cold weather. Greater amounts of cholesterol would be present to counteract the cold temperatures, which the cold temperatures make the bilayer more solid. So to counteract that, you make it more fluid. Proteins. Proteins are another, can also be found in the lipid bilayer. There are two main categories of proteins which are found in it. There is integral membrane proteins, which go through the inside of the bilayer. The only way to remove them would be to destroy the bilayer, and there's two types, transmembrane proteins and lipid-anchored proteins. Then there's peripheral membrane proteins, which are only found on the outside of the bilayer, and they don't interact with the hydrophobic inside of the bilayer. Membranes have two main functions. One is membrane transport. While some molecules can pass directly through the lipid bilayer, others need proteins. They could be used in either facilitated diffusion, which is diffusion with the aid of a protein, or active transport, which is when substances or molecules need energy to move up a concentration gradient. And also cell adhesion, that allows two cells to join together. Proteins play a large part in that. So finally, carbohydrates. They are attached to lipid bilayers through a process called glycosylation. When a carbohydrate is attached to a lipid, it makes a glycolipid. And when a carbohydrate is attached to a protein in the lipid bilayer, it makes a glycoprotein. Carbohydrates mainly play a role in cell-to-cell recognition. Okay, so permeability. This is permeability with respect to the plasma membrane. Some molecules are small enough to pass through the bilayer without any other forces, like gases, for example, or ethanol is another example, because it's small and uncharged. But molecules that are larger, ionic, or hydrophilic cannot pass through by themselves. Large molecules cannot go through the heads of the phospholipids because they're too large. Simple. And ions and polar, or hydrophilic molecules are repelled by the hydrophobic interior. Okay, so now we'll get to diffusion. Diffusion is the movement of a substance from high concentration to low concentration. These areas of concentration are called concentration gradients. It is a type of passive transport, meaning that it does not need an energy input. Proteins can play a role in diffusion, which is facilitated diffusion, which I explained before. Cells, what they do with diffusion is they try to keep a constant internal environment. But they never want that environment to equal the outside of the cell. Because if this environment reaches equilibrium with the external environment, it's a dead cell. Excuse me. So types of diffusion. There's diffusion. There's regular diffusion. I have a picture here, where molecules are just pass through the lipid bilayer without a problem. Then there's facilitated diffusion, which again is diffusion, which is still from high concentration to low concentration, but with a protein. And active transport is in this picture, but that is not diffusion. So the purpose of this lab, or this experiment, was to recreate a cell and its extracellular environment and determine the solute movement. And we did this by adding a solution to dialysis tubing, which was the cell, and placing it in a beaker of extracellular fluid. The cell contained a solution of starch and Na2SO4, or sodium and sulfate, and the extracellular fluid contained albumin, which is a protein, and chlorine. So our hypothesis was that starch and protein would stay in their environments, but sulfate and chlorine and the chlorine ions would move from one environment to another. So the procedure for this lab, first you had to set up the cell and its extracellular environment. So first you would close off one end of the 25 centimeter dialysis tubing. Next you would fill it with 25 milliliters of the starch and Na2SO4 solution. Then you would close the other end of the tubing using the clips. Then you rinse the bag with deionized water and pat it dry. Then you place the bag into the extracellular fluid, which is the albumin and chlorine. Then you start recording time, and you allow the experiment. We let it run for 45 minutes. So next you have to determine the direction of solute movement. So first you had to label 8 test tubes, 1 through 8. And once the 45 minutes has ended, you poured 25 milliliters of the solution in the beaker into a 25 milliliter graduated cylinder. That was the extracellular fluid. In test tubes 1 through 4, you would add 5 milliliters of that solution. Then you would clean the graduated cylinder and repeat that for the intracellular fluid, but use test tubes 5 through 8 to separate them. I'm sorry. This should be over here. First you would cut open the dialysis bag, then you would empty it into the graduate cylinder, and then you would repeat steps. And then you would do step 3, which is add 5 milliliters of the solution to test tubes 5 through 8. Next, you had to do some tests. First, you would test for starch in tubes 1 and 5 with the Lugol solution. If starch is present, the solution would turn a dark blue to black. Then you would test for sulfate ions in tubes 2 and 6 with 2% barium chloride. If sulfate ions are present, a white precipitate will form. You would test for chlorine ions in tubes 3 and 7 with silver nitrate. If chlorine ions are present, a milky white precipitate would form. And then number 9, you would test for protein in tubes 4 and 8 with a biurea reagent. If protein was present, the solution would turn a light lavender. So here were the test tubes before we added the agents. This is the extracellular fluid, and this was the fluid inside the cell. Here we had Lugol's iodine in test tubes 1 and 5. As you can see, the extracellular fluid, it did not have any starch, but the inside had a very high concentration of starch. So next, I'm sorry, this is hard to read, but next we tested test tubes 2 and 6, adding 2% barium chloride to test for the, excuse me. Here we were testing for sulfate, and if sulfate was present, it would be a milky white or a white precipitate. As you can see, both of them had it, but the fluid inside the cell had a higher concentration than outside the cell. This was for test tubes 3 and 7, after we add silver nitrate testing for chlorine, the chlorine ions. As you can see, they both tested positive, but in this case, the extracellular fluid had a higher concentration than the fluid inside the cell. Here were test tubes 4 and 8 after adding the biurea reagent, testing for proteins. This fluid inside the cell did not contain any proteins, but the fluid outside the cell, as you can see, because it's a lavender color, did contain proteins. So the final results for the starch test, test tube 1, it turned orange, which was negative. Test tube 5 turned to black, which is positive, it had starch. The sulfate test, both test tubes tested positive, but test tube 6 had a higher concentration. For the chlorine ion test, again, both tested positive, but this time the fluid that was outside the cell had a higher concentration than the one inside the cell. And for the protein test, only one had it. That was the fluid outside of the cell, and the other one was clear, so it did not have any proteins. So our conclusion would be starch was found in the solution containing the intracellular fluid, but was not found in the extracellular fluid. This was because starch did not diffuse across the membrane because it's a very large macromolecule, and the membrane wouldn't allow it. Sulfate was found in both solutions, but more so in the intracellular fluid. This is because sulfate was small enough for some of the molecules to pass through, but not create equilibrium. The same goes for chlorine ions. Chlorine ions were also found in both solutions, but more so in the extracellular fluid this time. Chlorine was also small enough for some of the ions to pass through the membrane, but not create equilibrium. Protein was present in the test tube with extracellular fluid, but not the test tube with intracellular fluid. This is because albumin, which was the protein in the extracellular fluid, is a large macromolecule and is unable to pass through the membrane. And that is the end of part 1 of our presentation.